In the Joglekar lab, we study one of the most fundamental processes necessary for all life, cell division. In particular, we focus on the step that ensures that each cell inherits an exact copy of the genome, the segregation of chromosomes. To segregate chromosomes properly, cells have evolved a sophisticated multi-protein machine that assembles on every chromosome called the kinetochore. The kinetochore performs several functions, including binding to spindle microtubules, and it achieves all these functions using close to 100 different proteins, all present in multiple copies.
By studying the kinetochore, we're really studying how cells ensure that they make two exact genetic copies of themselves. If a cell loses or gains chromosomes during cell division, it can lead to many devastating effects, including developmental diseases, cancer, and cell death. To prevent errors in segregation, the kinetochore must perform with high fidelity throughout the trillions of cell divisions that occur during the normal human lifespan.
The kinetochore has three main functions that help it achieve proper segregation. First, there is its ability to generate force. The kinetochore does this by binding to spindle microtubules and harnessing the energy of their depolymerization. This allows the kinetochore to align chromosomes and segregate them into daughter cells.
The second main kinetochore function is the spindle assembly checkpoint, or the SAC. When the kinetochore isn't bound to any microtubule, it generates a signal via the SAC to pause the cell cycle. This gives chromosomes time to attach to spindle microtubules and avoid missegregation. As kinetochores bind microtubules, the SAC is turned off and chromosome segregation proceeds.
The final kinetochore function is its ability to correct errors in microtubule attachment. Occasionally, kinetochores bind to microtubules in a way that will lead to missegregation if left unchecked. Fortunately, the kinetochores specifically detects and destabilizes these bad attachments, allowing them to reform their attachments correctly.
All the kinetochore's functions are aimed towards the same goal, accurate chromosome segregation. The lab is addressing several questions regarding kinetochore function and how it influences chromosome segregation. Here are a few of the projects ongoing in the lab.
One direction of the lab is to understand the precise mechanics of the SAC. How does a single kinetochore produce a signal strong enough to pause the cell cycle when only one chromosome is unattached? How is the strength of the SAC affected in cancer cells? By understanding the inner workings of the SAC, we are getting at the root of chromosome segregation failures with hopes of fixing it.
A second direction for the lab is to reverse engineer the kinetochore and build our own. We are using a rationale design to express and purify novel proteins which will assemble into the machinery for chromosome segregation. In other words, we are building a synthetic kinetochore with the potential for added features. Our ultimate goals are to better understand how the kinetochore works by trying to replicate it and use our synthetic kinetochores to control the segregation of any piece of DNA, natural or foreign, within the cell.